Hello everyone. Today I will discuss one of the important topic of CSR net syllabus, that is in developmental biology. It is a unit 5 that deals with basic concept of development. Now, today we will discuss the basic concept of development. One of the important part, that is specification, commitment, specification, and determination. Now, we know that when sperm and egg fuse, they form zygote, and the zygote divides and in the process of cleavage, and during cleavage process, it forms morula, then blastula, then gastrula, then organogenesis occurs, right? Now, we know that in commitment stage there is a early stage of commitment is called specification, and determination is the final stage of commitment. Now you can consider that here cell A and cell B. Now cell A in the early development, it will produce ectoderm form, and ectoderm, from ectoderm it will produce nervous system, and in B cell from mesoderm it will produce bones. So they both cell A and cell B have the equal potency, and when they present in neutral environment, they will one cell A produce nervous system and cell B produce bone cells. Now we'll see what do you mean by specification. What is specification? Now what is specification? Now see that the fate of a cell or tissue is said to be specified when it is capable of differentiating into a specific type of cell autonomously in a neutral environment where there is no competition or unfavorable components. That's called specification. Now in specification, always in neutral environment. Neutral environment means there is no competition, there is no unfavorable situation, and in a natural neutral environment, that is the neutral environment. Now suppose if a cell A, if you put undifferentiated cells, it's cell A, it will produce ectoderm when you put is neutral environment, but if you put in non-neutral environment it will produce endoderm. That means the fate of the cell can be changed. So specified cell can change its fates, but when a cell become determined, its fates cannot be changed. Now you can see the characteristic features of specification. It is the early stage of commitment. Second one, this phase is called labile phase, and it is a reversible phase. And if we change the cell position or environment, cell fate can be changed. Now if we put, suppose in B cell, that is in neutral environment, it will produce ectoderm. It is specified for ectoderm formation. Now if we put B cell in a non-neutral environment, it will produce endodermal cells, right? Now second, we'll see determination. What is determination? Now, a cell or tissue is said to be determined when it is capable of differentiating into a specific type of cells autonomously, even when placed into another region of the embryo or in non-neutral environment, that is modified medium or unfavorable condition. Suppose if you see that in a cell A here, now if you put neutral environment, it will produce ectoderm formed. Now if you put non-neutral environment, that it will also produce ectoderm. That means cell is determined for the ectoderm. Now so cell of another region of the embryo, suppose if you put the non-neutral environment, like you can, and you can give myo D signal. Myo D signal is a for mesoderm formation or myoblast differentiating factors which help to produce muscle formation. So cell is determined, that is its fate cannot be changed. So cell fate cannot be changed. The determined cells, it is a characteristic features of determination is final stage of commitment. It is, this is irreversible phase, and cell fate cannot be changed if we change cell position. So determined cells is a permanent cells, and it will not change its fates. Now see that, so first is the development is the commitment. So commitment, then it will produce specified specification, then the determination, then differentiation. So final stage is differentiation, before that determination, before that specification, and ultimate is the commitment. So commitment, then specification, then determination, then differentiation. Now we can classify the types of specification. How many types of specification are there? There is three types of specification. One is called autonomous specification, second one is called conditional specification, and third one is called syncytial specification. Now autonomous specification, auto means self. So autonomous specification here, the basically found in invertebrates, annelids, tunicates and molluscans. The autonomous specification we found, and conditional specification mostly found in the vertebrates like amphibians, reptiles, birds, and few invertebrates. And syncytial specification we found in the insect group, in between in the insect group. So autonomous specification. Now we'll see the characteristic features of autonomous specification. Predominates in most invertebrates. Specification by differential acquisition of certain cytoplasmic molecules present in the egg. Produce mosaic development. This is very important for exam point of view. This produce mosaic development. Mosaic development. Autonomous specification produce mosaic development. Conditional specification predominates in vertebrates and a few invertebrates. Specification by interaction between cells. Conditional specification is the ability of a cell to achieve their respective fate by interaction with other cells. Actually, these conditional specification cell conditional specification is the ability of a cell to achieve their respective fates by interaction with other cells. Interaction with other cells or a nearby cells. Actually, they secrete some paracrine factors. Actually, here, what a cell become is in large measure specified by the paracrine factors secreted by its neighboring cells. So another is the conditional specification leads to regulative development. Autonomous specification leads to mosaic development. Conditional specification leads to regulative development. Now specification by basically this type of conditional specification predominates in vertebrates and few invertebrates. Now syncytial specification predominates in most insect class. Specification of the body region by interaction between cytoplasmic region prior to cellularization of blastoderm. Syncytial specification basically predominates in most insect group where we see the multinucleated condition, and there we will find the syncytial specification. Now we can see here some experiment in early blastula stage specified cell stage, late gastrula stage determined cell stage. Now here is a donor cell, this is a host region, and cell fate. Cell fate is the ultimate the fate of the cell. Now if your donor cell is ectoderm, host region is endoderm, and it is sulfate is we found the sulfate is ectoderm. Now what's the donor region? Ectoderm. In if the host region is mesoderm, sulfate is ectoderm. Ectoderm is donor region. If the host region is neuron cells, it will produce ectoderm. See that here the fate of the cell is almost equal wherever the donor cell is, right? Look at that, the donor cell, and look at the cell fate. Whatever the host region is, it don't able to interfere with the donor host region cannot interfere with the donor cells. So here donor cell fate is same as transplanted cells, so there is no role of host region here. So here we can say that prospective potency equal to prospective fate, right? So next is, you can see that donor cell, host region, cell fate. Now donor cell is ectoderm, host region is endoderm, cell fate also endoderm. Ectoderm, donor cell ectoderm, host region mesoderm, cell fate mesoderm. See that donor cell ectoderm, host region neuron, and cell fate neuron. Whatever the host region is there, cell fate is determined by the host region. So here we can say that prospective potency greater than prospective fate, means prospective potency of the cell greater than prospective fate. This can be explained by the one is called Derich pressure plate experiment. Derich, this can be explained by the Derich pressure plate experiment. ESH, Derich pressure plate experiment. Pressure plate experiment experiment explain this procedure, explain this fact, explain this fact, right? Fact means here the prospective fate greater than prospective prospective potency greater than prospective fate. Now, next we'll see the differences between autonomous, conditional, and syncytial specification. Now, signal autonomous specification within autonomous specification within the cell cytosol, signal present within the cell cytosol. In conditional specification from neighboring cells or surrounding cells. Syncytial specification gradient of morphogen in cytosol. Now, signal in the forms of morphogenetic gradient, cytoplasmic determinants, mRNA, and protein. Conditional specification based on cell-cell interaction such as paracrine signaling, juxtacrine signaling are there for conditional specification. But in case of syncytial specification, morphogen gradient play important role in syncytial specification. Morphogen gradient, what is morphogen? Morphogen is a diffusible biochemical molecule. Next, in our next lecture, we'll discuss the morphogen. What is morphogen? How morphogen play important roles? So morphogen gradient play important role in syncytial specification. Now, suppose in case of Drosophila development in anterior region bicoid present, posterior region nanos. So anterior posterior determination of the Drosophila embryo, anterior region and posterior region is determined by bicoid in the anterior region, bicoid and nanos in the posterior region. So these are actually morphogen gradient play important roles. Specification early 8 cell stage, this conditional specification mid blastula stage, and syncytial specification syncytial blastoderm. Determination mid blastula stage, cellular blastoderm formation. Potency of the cell, prospective potency equal to prospective fate, prospective potency greater than prospective fate. Cell fate change or not change. So not change can, because it is a autonomous specification, conditional specification can be changed, cell fate can be changed, and it leads to mosaic development autonomous specification, and in case of conditional specification leads to regulative development. Conditional specification leads to regulative development. So in our next discussion we'll discuss about stem cells property and induced pluripotent stem cells, and then we'll discuss the morphogen gradient in the how the morphogen gradient play important role, what is the characteristic features of morphogen, and the French flag analogy model of morphogen, how morphogen play important roles in development. Hope this make helpful. Thank you.